Okay na ata. May bago na ulit. Okay. So, we have a new one. Alright. So, number 34. Okay. So, it's a number 34. I just moved it. Is it going? Is it continuing? Patrick?
Ano sir, Bacteriology 2024. Nasa ano pa rin sir,
Ayan na.
number 34. This is the structure of in some bacteria pa rin yung first.
Ay, nakita ko na dito. Ayan na, nagbago na. Oo. Nakakaloka ka, Patrick. Ikaw ang may delay. Char lang, Patrick. Ano ka? Niloloko lang kita. Char lang. Alright, let's continue the class. Okay, so we're talking about the important structure that is found in, medical important bacteria that constitute spore formers. No, that's not the one that we were talking about earlier. This one. So the structure that is found in organisms that is considered, or that is also known as a resting cell, and allows the organism to survive under extreme condition is a spore or an endospore, to be specific, because we have spores in mycology, so I don't want you guys to be confused. So endospores, these are found within the bacterial cell. And the components include calcium dipicolinate or dipicolinic acid. Okay? That's the answer to that one. Now, we move on to the medically important bacteria that are spore formers. You only have two. Can anybody name it very quickly? Let's go with Peter John. Peter John, medically important spore formers. Do you know? Ay, wala si Peter John. Sige, si Arian Dorothy. Sige. Kasi nakikita ko si Arian Dorothy. Nawawala si Marian. Ano to? Nag-break si Marian.
So, nandito ako.
Nandiyan ka. Hindi ko nakikita si Arian muna. Sige. Medically important bacteria that are considered spore formers.
Circlostridium po. And Bacillus.
Okay, very good. So the location of the spores in these organisms, it's central for Bacillus and it's either subterminal or terminal for Clostridium. Okay? So that's what it looks like. For Clostridium, all species of Clostridium have a subterminal spore, except for Clostridium tetani, okay, which is terminal, making it look like it has a drumstick appearance. Bacillus, on the other hand, all of the species that we're going to talk about have a centrally located spore, as depicted in that drawing. Ang pangit yung drawing, pero you get the idea. Okay? Alright, ang pangit yung drawing, no, Patrick? Okay, let's move on to the next slide, which is number 36. Okay, so number 36, examples of stains used to demonstrate endospores. We have three. Under those three, we have the Schaefer-Fulton stain, Ritz-Conkin, and Dorner stain. Sir, ang dami naman namin na-aralin ni. Meron pang histopat. Sir, chill lang kayo, class. Hindi lang yan na-aralin yung marami pa. Okay, so we have Schaefer-Fulton, Ritz-Conkin, and Dorner. Okay, and the endospores are stained differently, or are stained with different colors depending on what type of staining technique you use. Endospores are stained green with Ritz-Conkin and Schaefer-Fulton, and Dorner stains it red. Okay, stains the spores red. Okay, Ritz-Conkin and Schaefer-Fulton, green, Dorner stain, red. Okay, now let's move on to the structures that may be found in the external cell wall of bacteria. We have two things that you need to remember. You have a capsule and a glycocalyx. Okay, or sorry, a slime layer. Sorry. So it could be a capsule or a slime layer, which is outer wire, and then a glycocalyx. So any of the two. Okay, the function of capsules and the pathogenicity of microorganisms is number 39. What is the function of capsules in the pathogenicity of bacteria? It inhibits phagocytosis. Okay, as I mentioned before, what are the three organisms that we were talking about that had capsules? Ritz-Conkin, yeah, Ritz-Conkin, not Ritz-Conkin. Ritz-Conkin. Yeah, I forgot to change that one. Okay. Thank you, Sir Marco. Was that Sir Marco? Yeah. Okay. So, what is the capsules that? What are the organisms that have capsules? These are causative agents of what do you call this? Bacterial meningitis. So, what are the three? What are the three ones? Recall tayo dun sa pinag-usapan natin kanina. Nasaan na si Marian? Si Marian yung pinag-chismisan namin ni Patrick. Kunyari magpo-intern kami ni Patrick. Ganun, Patrick, alam mo pa. May jowa na sige, iba naman. Hindi na nag-response si Marian. Iba naman. Si Arian Dorothy naman. Arian Dorothy, what are those three organisms that we talked about earlier that had capsules? Forgot na class. Guys, you forgot na Haemophilus influenzae, Neisseria meningitidis, and Streptococcus pneumoniae. They are encapsulated. Okay? There are three general methods, by the way. I think that's number 40, right? Di ba talaga, memorize ko pa talaga yung lecture ko yun. Pinamove ko lang kay memorize ko pa talaga siya. So there's three general methods in demonstrating bacterial capsules. For number 40, the answer is you either use a milk media, or you use staining, or you do antigen stereotyping. So milk media is fairly easy. Anything that grows on milk media would probably be an organism anything that grows on a specific milk media. We use a different milk media. We'll talk about that later when we go to culture media. But suffice to say, there's what we call a capsular milk media that we use in the lab. Staining using what? Using a capsular stain or a background or relief stain. And when we're using antigenic stereotyping, we're targeting the antigens, which is basically the capsule, making it undergo swelling. An example of this is Neufeld-Keelang test, or the Neufeld-Keelang test, or the capsular swelling test. So those are the three methods that you will encounter, or the three methodologies that we can use to demonstrate capsular structures in an organism. Okay, so let's start with number 41. Number 41, bacterial capsules can be stained with two things. There are two staining methodologies. You could use the HISS method or the HIS stain, or we could use nigrosine. Okay, nigrosine or /India ink. The reason why it's called the background stain or a relief stain is that you're not really staining the capsule. More so, you're staining the background, hence the term background stain. What happens is that when the whole background is black and you see distinct appearance of the yeast, or in this case, not yeast, or the capsular organism, because most of the time we use it on CSF. So that's why I said yeast earlier. So when you have a capsular organism, it will reject the stain it will reject the India ink or the nigrosine stain. Okay? But it will stain everything else. Okay? So if it is positive for a certain capsular organism, rest assured that it won't stain the cell that has a capsule, but it will stain anything else in the slide. Okay? So what else is there? I think in number 42, we're going to talk about the reagents used in the staining method or HIS. There are three things that you need to remember. One of those things is that you use gentian violet as your pre-stain, and then you have used basic fuchsin. Okay? So you combine these two things, and it will bind to the capsule. Okay? So gentian violet and basic fuchsin will bind to the capsule. The next thing that you're going to do is you're going to remove the metals using a copper sulfate wash. You don't use water for this stain, by the way. You use copper sulfate. You know that thing that you, if you rotate it in the blood bank, you know, it's usually in a beaker. And then if your donor has 12.5, if your donor has less than 12.5 has less than 12.5 grams per deciliter of hemoglobin, it will float. And then if it's more than, it will flow, and then you accept the donor. Do you guys have that when you were in blood bank? Or do you have the POCT or the point of care hemoglobin testing method na? Social na ang labs ngayon. Or you don't test the patient, you don't test the patient's hemoglobin or the donor's hemoglobin. Yung copper sulfate na yun, that's the blue solution that we use. And it's even the same thing as the thing that you're using for Benedict's test when you want to test for calcium, for sugars in the urine. Okay? So that's what we use with HISS stain or HIS stain. Okay? Now, we're go to the next slide. I think the next thing that we're going to discuss is the serotyping technique used to demonstrate the presence of a capsule in some bacteria. Again, as I mentioned, I said some bacteria, okay? So the antigenic serotyping technique is known as the Keeling test, and the positive result for that one is capsular swelling. You actually induce the cell to swell up, okay? It swells up, lumolobo siya. Okay? If it's positive for that one. It's known as the Neufeld-Keeling test. Principle of which is that you activate the complement system, making it explode in the process. Okay? Now, this is a nice to know question. I think this was a question. The next one, I think is 44, right? I think we're in number 44. The slime layer is observed in what species of Staphylococcus? Okay? So who here can answer that question? Marian Vanis Castro.
Ano po, sir? Staphylococcus epidermidis.
Very good. Nakakatuwa talaga. Alam pa nila. Okay? Staphylococcus epidermidis. Alright. Let's go to the next one. Capsules in some bacteria are also synonymous to what antigens. So please don't forget these terms, the K antigen or the VI antigen. Okay? K antigen for most types. Okay? And VI antigen is actually for Salmonella typhi. Okay? K antigen for most types. VI antigen for Salmonella typhi because the capsule for this organism usually has another component in it. Okay? It's not like the other one which is made of polysaccharides. Okay? Next, we have another structure to talk about. This is now the number 46, I think. Yeah. Okay? Number 46. Based on function, what are the two types of pili structures in bacteria or pili? Pili. Hindi pili yan ha. Hindi yung pili nuts, okay? Patrick, nagugutom ka na ha. Hindi pili nuts yan. Patrick, nako. May pili. Gumanon. Yung itsura ni Patrick, gumanon talaga siya sa camera. Hindi yan pili nuts ha. Kakaloka ka, Patrick. Hoy! Anong oras na nga ako dumating dito? May break. 1 o'clock tayo mag-break. Usog natin ng ano. Anong oras ba dyan? Alexa, what time is it in the Philippines? Tumigil ka, 1 o'clock tayo ha. Kakaloka ka, Patrick. Kakaloka ka, Patrick ha. Pili nuts ka agad. Late na nga ako dumating dito eh. Kayo talaga. And that was approved by the dean. Okay? Hindi nyo pwede. Si Dean na makapangyarihan sa atin. Ako'y nagre-request lang. Okay, natawa ako kay Amanda. Natawa talaga. Ayaw ko, hindi ako maniwala sa inyo kasi baka mamaya i-scam nyo ako. Kaya Alexa, ako maniniwala. Niloloko nyo. Baka mamaya niloloko nyo kaya na-etchos nyo ako eh. Alright. Let's go to the next. The based on function. Yeah, so okay, we have two types. We have the ordinary pili and we have the sex pili. Okay? So let's talk about the ordinary pili first. Or the reason why it's capable of adhering to the host cells is because of this ordinary pili. It's also known as the adhesion pili. Alright? Okay? And the sex pili, sir, may nagsisex pala ang mga bacteria, sir. Yes. Okay? So yung pili na yan is basically as you would use it as a human. It is to transmit the genetic material to another cell. Okay? So ganun nila ginagawa yung kabulastugan nila class. Okay? Using a sex pili. Okay? So one So there's no male and female, of course, right? It's just in some organisms they just need to complete the mesosome. Okay, so for this type of bacterial conjugation, or for bacterial conjugation, in order for a cell to undergo binary fission, it needs another copy of the genetic material, and that is through the use of the sex pili. So parang ini-inject mo yung genetic material, which is basically what other organisms in the animal kingdom does. It just so happens that it's not for pleasure. Okay. All right, let's move on to the next slide. The next slide talks about stains in determining bacterial flagella. So we're talking about bacterial flagella. Before we continue about stains demonstrating flagella, I want to know if the class understands the function of a flagella. Anybody know here? Anybody here can answer what the flagella is for? What is the function of a flagella? Para saan ba ang flagella? Okay. Sige, magtatawag na lang ulit ako. Si Shaira naman. Kasi si Shaira ay parang ganyan yung itsura ni Shaira. Hindi ko alam kung ano tinatay ni Shaira, pero sige, parang feeling ko about flagella yun. What's the function of a flagella?
For movement po, sir.
Very good. Or motility. Okay? Motility. Very good. Okay, so for movement or motility. Grabe naman yung movement. Pag sinabi kasi ni Shaira na movement, parang yung naglakad from EDSA Caloocan to Roosevelt Munoz, no? Parang ganun yung pagkasabi ni pagka sinabi niyo for movement, motility type. For microorganisms, we call it motility. But that's also the same thing. It just so happens it gets a little bit fuzzy when you use it in terms of in the perspective of microorganisms. But anyways, we can stain these structures. By the way, class, baka lang kalimutan ninyo. We can use what? We can use what type of stains? Escher-Con na naman, hindi. Char lang. Puro kayo Escher-Con. Leafson stain, Gray, and Escher-Con. Okay? Escher-Con. Ritz-Conkin yung nasa isip ko kanina. Wirtz-Conkin. Mali ako ng spelling class. Sorry naman. Okay? And what particular component or reagent do these reagents have in common? They all have tarnic acid. Okay? And in their exams, hindi isa-isahin class na naman, ano yung ginagawa for Leafson stain? Ano yung ginagawa for Grayson stain in Escher-Con? What they ask in the exam is, what component is important when technologist wants to target the flagella? And it is usually tarnic acid. Okay? Ang dami nyo ng klaseng acid na tatandaan. Teichoic acid, echoic acid, mycolic acid, di ba? Ang dami, no? Muraric acid, di ba? Ang dami nyo kaalala ng mga acid class. Kaya, huwag kayo mag-alala, dagdag ko pa yung class. Okay? Next. Bacterial flagella. There are several types of bacterial flagella. Last year, nagkakagulo-gulo yung mga students kasi kung saan sila nag-google. This year, hindi ko na sasagutin. Hindi ko na pa sasagot sa kanila kasi naloloka sila. I will answer it by myself na lang. We have an atricus flagella, or structures ng flagella muna tayo. What are the types of bacterial flagella? There are 6 types. Okay? Atricus meaning no flagella, monotricus, mono, 1, amphitricus meaning 2 or both poles or both ends. And then we have a Loprotricus flagella. Pag sinabing Loprotricus, merong tuft of flagella, meaning pag sinabi natin tuft, there's one point, but there's a lot of flagella originating from that one point. We have peritricus flagella from the term itself, perimeter, ibig sabihin all over the organism, and then periplasmic flagella or axial filaments, which is the one that you see in spirochetes. Okay? Clear ba tayo doon, class? Isa, both ends, one side, pero maraming lumalabas, may tuft or parang pom-poms. Okay? Peritricus all over the organism, or perimeter, the perimeter of the organism, and then periplasmic flagella or periaxial or axial filaments, which is the one that you see in spirochetes. Okay? Now, what is the function of flagella? Sinagot ko na yun kanina, di ba? What's the function of flagella? I think that's number 49, right? We're going to the next slide na. Patrick. Nagugutom na talaga siya dahil sa pili nuts. Okay? So the function of flagella is for, as what Shira answered earlier, it is for motility or movement, which is best observed at room temperature. Okay? Ganito lang yan, class. Ang mga mikrobyo, gusto lang nila yung chill lang ang buhay nila. Okay? Hindi sobrang chill, yung chill lang ang buhay nila, class. So, mga room temperature lang tayo. Bakit? Pag tinanong kayo class sa board exam, which of the following temperatures would most likely induce or reduce the motility of the organism? Heating the sample, subjecting the sample to room temperature, or freezing the sample, or all of the above. Siyempre, ang isasagot, or both A and C, mga ganun yan, would impede the movement. Ibig sabihin, impede, it will stop the movement, or it will interfere with the normal function. Diba? So, pag sinabi niya, pag ganun ang mga tanungan, dapat maintindihan nyo na chill lang ang buhay nila. Gusto nila normal temperature lang, room temperature lang. Or we call it ambient temperature. Ang arte dito sa US, ang sabi nila, ambient temperature lang. Arte, di ba? Okay? So, room temperature. So, what's room temperature? 25 degrees Celsius, di ba? So, it's best observed at 25 degrees Celsius. Okay? Now, let's move on to the next slide, number 50. I think it has something to do with organisms that become immotile at 37 degrees Celsius. These include your Yersinia species and Listeria monocytogenes. Okay? Organisms that are inhibited from their motility function or their flagellar function at 37 degrees Celsius. This includes Yersinia pestis or Yersinia species and Listeria monocytogenes. Okay? Now, Peritricus flagella is number 51. I think that's for number 51. Teka lang, sagutin muna natin yan. Number 50, Yersinia species, 50A, sorry. Yersinia species, Listeria monocytogenes. Okay? Now, we go on to number 51. Peritricus flagella is observed in all species of the Enterobacteriaceae family. By the way, class Enterobacterialis na siya. Ayaw na nilang mahirapan yung mga medtech. Okay? Just in case marinig niyo yung term na Enterobacterialis. Okay? That is nahirapan kasi yung mga bagong microbiologists ata. Nahirapan silang i-spellin yung Enterobacteriaceae. Okay? So, ginawa na lang Enterobacterialis. Okay, so the contemporary nomenclature for the Enterobacteriaceae family is now known as Enterobacterialis. And all of them have Peritricus flagella except for Klebsiella species. Okay? Klebsiella species. Okay? Bakit? Ano ba ang klase ng flagella meron ng Klebsiella? Ano ba? Atricus, monotricus, peritricus, amphitricus, axial filaments ba ang meron siya? Ano? Bigyan ko kayo ng clue. Ang imbig result niya is negative, negative, positive, positive. Bigyan ko kayo ng clue. Indol, methyl red, Voges Proskauer, ang ibig sabihin Voges. VP na lang, Voges Proskauer, kasi nahihirapan ako i-spell si Proskauer. And then, citrate. Sibon citrate. Si Klebsiella, negative siya. Negative, positive. Bakit? Ano bang meron structure meron siya? Number 40 na tayo. 52, sorry. Number 52 na tayo, Patrick. Mamaya natin sagutin yan. Question ko is, what type of flagellar structure does Klebsiella species have? Kaya natin. Given that you have these results. Class, baka mamaya itanong ko yan sa board exam. Or mamaya itanong ko yan sa exam ninyo. Ano ba dyan ang test for motility? Marian, ano dyan ang test for motility? Dun sa apat na nilagay ko sa chat box.
Wait lang, sorry.
Sige. Bigyan kita ng time para mag-Google. Sige, hanap ko na ako ng ibang classmate niyo. Si Arian Dorothy.
Sir,
Ay, ayan na tayo. Sige, mamaya na Arian.
hindi.
Si Marian muna. Sige, Marian.
Flagella,
What type of
Sasagutin.
flagellal structures? What type of flagellal structure?
yung one pole, sir.
Bruha.
Um,
Wala siyang flagella. Kasi bakit? Negative yung indol niya. Ibig sabihin, no motility. Okay. Okay. Kung meron. Ay, ganun pala, sir. Ginugol ko. Bigay ka sa akin ng Klebsiella species na ano. Sasagutin kita, hindi yan Klebsiella pneumoniae, hindi yan Klebsiella rhinoscleromata. We're talking about this medical microbiology, okay? Iba yan siguro nakita mo yung may one pole na yan. Last year kasi ganyan din na tinanong, sinagot ng isa kong student. Pinabasa ko sa kanya kung ano Klebsiella species yun. Hindi yung topic sa my medical microbiology. Okay? So, for Klebsiella pneumoniae, rhinoscleromata, and Klebsiella aerogenes, they are atricus. Okay? So they don't have a flagella. Okay? So let's go on to flagella is observed. What type of flagella is observed in Vibrio species or Vibrio species? Ano nga ba, class? What is it ba? We have what? A monotricous flagella. Isa lang. Okay? Monotricous flagella. So class, huwag yung makalimutan yung importance at saka yung relevance ng mga English result. Baka mamaya kasi memorize na kayo lang memorize, hindi nyo naalala, di ba? Ano bang ginagamit natin na media for indole test, di ba? Sulfide indole motility, di ba? Kaya nga SIM ang tawag yun, semi-solid na media siya, di ba? Okay? Now, examples of test for motility. So, nasagot yun na yung isa. Diba? Number 53 na tayo, Patrick. Examples of test for motility. We have the hang and drop method. Let's show the answers also. We have the hang and drop method. We also have the stains for the flagella and the semi-solid medium, which is what we usually do, what we usually use in, utilize rather, in the clinical laboratory. Okay? Now, let's talk about, I think, certain organisms and their motility movements. Bakit kailangan alalahanin? Kasi every time na nakikita ko sa board exam, nandito siya lagi. Okay, for Listeria monocytogenes on a McBride media, it has what you call a tumbling motility. So, I think it was from Legend Review Center that I got this mnemonic from. Mapapatumbling ka sa sobrang anghang ng Listerine. Diba? Hindi ko alala kung sino yung nagsabi nun, si Dr. Arian. Basta sa nanonood kasi ako ng isang lecture sa review center. Siyempre para mangopya sa kanila. But I'm still giving them a shoutout. Baka mamaya sabihin na nanakaw ako ng ideas nila eh. So, para hindi nyo makalimutan ang itsura ng motility or characteristic motility pattern ng Listeria. Tandaan nyo lang, pag nag-Listerine ka sa sobrang anghang, lalo na yung kulay green. Okay? Ay, lalo na yung kulay light blue, yung kulay copper sulfate. Sa sobrang anghang niya, mapapatumbling ka. Okay? Tumbling motility on McBride medium. Okay? Now, Caprocytophaga is an organism that we may not speak about, but it is under the HASEC group. Okay, the HASEC group includes Haemophilus aprophilus, Actinomyces, Actinobacillus, Mycobacillus. What else? The other one, HASEC C. Actinobacillus na lang. Baka nabago na eh, tsaka mali ako. Ano nga ulit yun? Haemophilus aprophilus, Actinobacillus, C. Ay, si K pala. HASEC. Letter E muna tayo. Echinella corrodens and then, ayan, Capnocytophaga pala. Capnocytophaga. Okay? And then, we have Capnocytophaga that has gliding motility. Then we have tumbling Campylobacter that has darting motility. Ano pinagkaiba? Gliding motility is yung parang pag nagsislide ka lang. Has anybody here seen somebody who watched the Olympics and they saw the ice skaters? Parang ganun yung movement niya. Dahan-dahan lang siya pero graceful. Now, yung darting motility, may stops siya in between. Okay? So, I would say the difference between Capnocytophaga and Campylobacter, when you look at them in the microscope, is like, my analogy with this one would probably be a professional ice skater versus somebody who's just learning how to ice skate. Yung isang pro, yung isa natututo pa lang. Bakit? Pag bago ka pa lang nag-aaral mag-ice skate, di ba? Dahan-dahan ka pa lang kasi pa stop ka. Pero since si Capnocytophaga, siya yung ayun, alala ko na. Kingela Kingay. Okay? So, gano'n. Yung pala yun. Okay? Nalito lang ako kanina. Okay, yun yung HASEC group natin. HASEC. Tapos bago na din ang name pala ni Haemophilus aprophilus. Agregobacter na siya ngayon. Agregobacter. Okay? Alright, let's move on to the next one. Please don't forget Listeria, tumbling, Capnocytophaga, gliding, and then Campylobacter, darting. Okay? Now, let's go to the most commonly used media for bacterial motility that's known as SIM media or the SIM media, sulfide ethyl motility. And based on composition, it is considered as a semi-solid medium. Okay? Semi-solid medium siya, class. Okay? Clear? Okay. So we have to differentiate also pathogenicity. I think that's the next topic, right? Do we have anything else? Let me just look at my notes. Okay, so we don't have anything else. I think we can move on to the next topic, which is pathogenicity versus virulence. Okay? Pathogenicity is different from virulence. Okay? What's the difference? Anybody? Anybody know what's the difference between pathogenicity from virulence? Well, the answer is pathogenicity is the ability of an organism to... Ang tagal ni Mario, may moyo eh. Moyo naman eh. Sinagot ko na. Okay, so for pathogenicity is the ability of an organism to cause a disease, and virulence is the degree of pathogenicity. Okay, so the degree at which that particular organism can... is it severe, is it moderate, or what degree is the disease? Or is the ability of an organism to cause a disease. Okay? So virulence, that's why we have virulence factor. Diba? So you remove one part of the one virulence factor, it becomes less pathogenic. Diba? Or it becomes less virulent rather, sorry. Okay? So let's talk about pathologic determinants. That is our next topic, I think, number 57, right? So pathologic determinants includes your adherence factors, your antiphagocytic factors such as M protein or mycolic acid. We also have enzymes and toxins. These are pathologic determinants. This is what determines whether or not an organism is capable of causing a disease. Let's say, for example, we have Staph aureus, and the other one is methicillin, and the other strain that you have is methicillin-resistant Staph aureus. That is considered. Which one do you think would be considered as pathologic or less pathologic than both? The one with the resistance factor, or the one that has a resistance gene, or the one that doesn't have a resistance gene? Ano ba yung MRSA class? Methicillin-resistant Staph aureus. That is a strain that is resistant to that is resistant to many penicillin-based antibiotics, which is what we're going to talk about next time. But I want you guys to remember is that whenever there is a resistance gene, something is much more pathologic. Okay, so we have also other things to take into consideration. Among the Clostridium species, which is the most deadly? Clostridium botulinum or Clostridium tetani? Or Clostridium perfringens or Clostridium difficile? Diba? So yon. So toxins sa pinag-uusapan naman doon, diba? So class, you have to remember pathogenic determinants also as part of our curriculum when we are studying microorganisms. Okay? So let's not forget. Let's show them again, Patrick. Let's show them, Patrick. Your examples of pathologic determinants include adherence factors such as your pili, antiphagocytic factors that includes your M protein or your mycolic acid, and then we have enzymes and toxins. Because we have a lot of enzymes that we're going to talk about later, I would rather we just discuss it based on organisms so you don't get confused, okay? Now, let's move on to number 58. What are the two types of toxins released by bacteria? And let's differentiate them based on the source. And when I say the source, whether is it gram positive or is it gram negative. So there are two types of toxins released by bacteria. We have, let's show them the answer, exotoxins and endotoxins. For exotoxins, it is released by both gram-negative and gram-positive bacteria. Endotoxins, on the other hand, are only released by gram-negative microorganisms. Okay? Endotoxins are basically part of the cell wall of these organisms, and therefore they can cause diseases. Okay? Exotoxins, on the other hand, are released by the organism. Okay? Kaya nga endo and exo. Endo, nasa loob or part ng organism. Exo, ibig sabihin they release it either as metabolites or waste products. Okay? Now, let's talk about the bacterial toxins based on molecular composition. When I say molecular composition, are they lipids? Are they carbohydrates? Are they proteins? Okay, so exotoxins can be let's go to the answer. 59A na tayo. Exotoxins can be protein or peptide, while endotoxins are more likely to become lipids. Okay? Specifically lipid A. Alright? Now, number 60. Differentiate the release mechanisms of exotoxins and endotoxins. Exotoxins are released by metabolic products or released through treatment, whilst endotoxins are released by the disintegration of cell wall or phagocytosis. Okay, so because it's already part of the cell. Okay, let's move on. Which bacterial toxin type is heat labile and which bacterial toxin type is heat stable? So let's go back to number 59, 59A. All right, and I want to ask the question, which of the following would most likely coagulate with heat? Exotoxin or endotoxin? Which one coagulates? Is coagulation a property of a lipid or a protein when introduced to heat? Marian?
Protein, sir.
Very good. So it is exotoxin. So let's go back to the answer. Let's see if we're correct. Tingnan nga natin kay Patrick. Okay? So heat labile ang exotoxins. So they're less stable when there's heat. Okay? When they're more stable, toxins are more stable when they are lipid based. So heat stable si yung endotoxins. Bakit, class? Kasi hindi naman mabilis masira ang lipids, di ba? Proteins on the other hand, when you expose not lipids, proteins to heat, they will coagulate. These things coagulate. They tuloy as if they're alive. Let's move on to the next slide. Number 62. Among all bacterial endotoxins, which of the following is not heat stable? So there is one exception, of course. Okay, siya lang ang bukod tangi. Okay, and this is the staphyloenterotoxin. Okay, this is the staphyloenterotoxin. Everything that I've talked about that are heat stable exotoxins are heat stable exotoxin, except for the staphyloenterotoxin. Okay, next, immunologic characteristics naman tayo. Based on immunologic characteristics, number 63, based on immunologic characteristics, differentiate exotoxins from endotoxins. What is the difference? Babalik tayo dun sa may biochemistry nila. Proteins versus lipids. So which one, immunology recall, which one is more immunologic, a protein or a lipid? Naku, nakalimutan na nila. Naku, patay tayo. Moyo. Hindi, kaya ano muna tayo, kay Moyo muna tayo. Baka sabihin meron akong favoritism. Kay Moyo muna tayo. Moyo, which one is more likely to induce an immune response? A protein or a lipid? Ayun na, sige na nga, hindi makatulog si Marian. Sige na, oh.
Sir, napipindot.
Ano na nga, Marian? Ikaw na sasagot, tinawag na kita eh. Wala na si Marian. Patay tayo dyan. Anybody from the class? Hala, wala silang performance. Patay kayo. Hindi nyo ba alam ang secret recitation? May secret recitation kami ni Sir Marco. Tinitignan namin kung sino yung nag-recite lagi. Sige, Marian.
Sir, protein ulit.
Protein. Okay, very good. Tama naman. Wala namang antigenic, therefore it can be neutralized by antitoxins. Okay? Endotoxins are less antigenic and therefore they're less likely to be neutralized unless of course they are lipopeptides. Okay? But anyways, let's talk about pharmacologic effects. Again, we're going to go back to the biochemistries. Which one is more efficacious? Which are the effects? So we have a cytotoxin, we have an enterotoxin, and we have a neurotoxin. So there are three things that you have to remember. A cytotoxin kills the host cells, an enterotoxin damages the cells of the gastrointestinal tract, and a neurotoxin interferes with nerve impulse oil transmission. I'd like to ask the class, can you give me an example of an organism that produces a neurotoxin, a potent neurotoxin? Ginga? Anybody in the class? A potent neurotoxin, actually 1 milligram of this 1 microgram rather, of this one, when injected through the bloodstream via IV, can cause death to a lab animal. 1 microgram. It's potent. James Paul.
Clostridium tetani.
You are in the correct family but wrong species. But yes, you are correct. They are capable of producing neurotoxins. The most potent, I'm asking the most potent neurotoxin. Moyo.
Clostridium botulinum po.
Very good. Botulinum. The botulinum or the botulinum toxin, or how we commonly know it now as Botox. Did you know that, guys? Did you know that Botox is actually from Clostridium botulinum? Di ba? Hindi niyo alam, di ba? Yung mga, wag kayo mag-alala, hindi pa ninyo kailangan. Yung mga itsura niyo, wala pa mga wrinkles. Pag nag-start na kayo magtrabaho, Arian at Shaira, humanda kayo. At saka ikaw, Claire Gono. Yung mga nakakita ko lang yung noo. Yun yung mga tsaka si Maria Tirlis. Yung mga ganyan. Yung mga lalaki, hindi na wala. Mas lalong umaasim ang mga lalaki pag nagkakakulubot eh. Alright, let's move on to the next slide, class. Number 64. Number 64 talks about toxicity of exotoxins from endotoxins. Exotoxins are highly potent. Exotoxins are highly potent, while endotoxins are less potent. Of course, so we've said that already, right? Let's move on to most potent bacterial toxin to man, number 66, I think. We've talked about that. The botulinum toxin comes from Clostridium botulinum. All right, we'll go to the next one. We want to increase the pace because we have limited time and we have to go through a lot of information. Okay, so endotoxins from body fluids and surgical instruments can be detected using amoebic lysate derived from one animal. Common name and scientific name. So common name, horseshoe crabs. And the scientific name is Limulus polyphemus. That's why we call it the Limulus test. Okay, it's horseshoe crabs and it causes clumping. Okay, so before any material from CSSD, or what we call the central sterilization and standard this department is sent to the OR, they would use Libellus polyphemus reagent. And Libellus polyphemus reagent is your quality control if there is clumping of the horseshoe crab lysate or the amoebic lysate. This would produce a positive. This would cause the rejection of the sterilized instruments. Okay. Now, that is the end of our first lecture for today. I think I have a playlist created for my 2022 class, and because this is an emergency, as I did this as a favor for Sir Marco, I wasn't able to change the dates. But I will see you this day. We'll just have like a 30-minute break. Will that be all right? Okay. So all lectures and discussion will be posted in the playlist. This is the same one, but if I'm going to make an updated version. And Patrick, I wish you would. I hope you would be as diligent as your predecessors, and we won't see each other next semester. Okay, but I will keep sending you the lectures for our next topic. Okay, so let me just start the clock so that we are in sync kasi meron tayong daylight savings dito sa US, kaya medyo nalilito ako sa oras. That's why I keep asking Alex. Wala akong tiwala sa inyo. Last time kasi ay hindi pala kayo yun, yung ko pala yun, ibang students ko pala yun. Mababait yung mga mababait yung mga nandyan sa CMCC. CMCC na ba or CMCC pa rin ba or CMCU na? University na ba or ano? Masyado na ako matagal na wala sa Pilipinas eh. Nasaan na yung alarm? Timer. CMCC po. Okay. Hindi ko alam, mamalay ko ba kung ano. Kasi masyado naman nagiging busy ang Sir Marco ninyo. Hindi ko na alam kung ano nang ganap. Okay, so ito yung timer natin, class. Kilala nyo naman ako. Gusto ko lagi strict and on time tayo lagi, class. Okay. I will stop